Okay, it looks like we have everyone joining in. So welcome everybody to, I believe this is our fourth session, if I'm correct, on FCS Express or FCS Express 7, our introductory course here. And my name is Kathy Daniels, if for anyone who hasn't joining us for the first time rather today. So just as a brief introduction, we've been doing these virtual classrooms on FCS Express 7 and learning the software. We've been lucky enough to have Sean Burke join us from DeNovo Software. So you'll see Sean there right below me. So thank you, Sean, for joining us. So just to give you a brief overview for what we're going to be going over today. For today in FCS Express 7, we're going to be reviewing the use of modeling in FCS Express for the analysis of cell cycle and for also looking at proliferation analysis and modeling. Sean's going to be going over a couple of different data sets, and specifically he'll talk about univariate and bivariate analysis for cell cycle and also looking at proliferation. So some important links as we go through. I just want everyone to be aware that if you already have FCS Express or if you're looking to download FCS Express, you can visit DeNovoSoftware.com. In addition to being able to download the software, you can also utilize that link for educational purposes, and they have a lot of information on their website. Then there's also the Flow Cytometry Core Facility website from MSKCC.org right below. We have plenty of content there, including all previously recorded sessions on FCS Express 7. So we have introductory, we have high parameter analysis, we have compensation and unmixing. So we have a wide variety of information and reportings on our website. In addition to that, we have our Twitter page, and our handle is @FlowMSKCC. We have our Flow Post-its that we post there, interesting articles, an entertainment series where we do some cover songs for flow cytometry, and we just posted one of those yesterday, Cytoman, so check us out there. We also wanted to make sure everyone's aware that there's availability through ISAC for education right now. They have made Cyto University available for free to all cytometrists around the world, as many people are working from home, so we thank ISAC for that initiative. And then flowrepository.org is available for any users who might be looking to do analysis with FCS Express 7 and don't have an availability of FCS files with them, or they don't have the FCS files with them to analyze. Feel free to go ahead and check out Flow Repository. Some more important links, Cytometry Part A is linked up top for if you're looking to get information on cytometry research and articles. We have that up top. Then there's our regional flow cytometry users groups, specifically MetroFlow, which is the New York, New Jersey, Connecticut. And I'd like to specify that we're going to be actually hosting a corporate member seminar series through MetroFlow that will take place on May 11th to May 15th. And more information on that can be found on metroflow.org. It's going to be 27 talks, including a talk from Sean, over the course of 4 days, and we're very excited for that. Okay, then we also have FlowTex, which is our partners down in Texas, and they've been amazing providing educational content while many people are working from home. So please go ahead and visit their website as well. And we also have a little YouTube link there to tell you how to go ahead and make that cool plot on that first page where we have the FCS Express 7 font turned into an FCS file, or text turned into an FCS file. Okay, so briefly before I let Sean get started, I just wanted to do a brief advertisement for our Flow Post-it program that I mentioned earlier that we post on our Twitter and on our website. So what you can see here, back in March, Barbara Oliveira from our group and myself put together Cell Proliferation by Flow Cytometry Post-it. And the idea of these is really to go ahead and provide very succinct but informative documents that we can send out so that everyone has an opportunity to learn a little bit more about flow cytometry as they're going about their busy day-to-day activities. So feel free to check out this cell proliferation post-it, and it goes over different methods to analyze cell proliferation by flow, some tips, and different dyes available and what they bind to within the cells to be able to see divisions within the cells. So please feel free to check that out, in addition to the many other flow post-its that we have available. Okay, so some contact information. Please feel free to reach out after today if you have any questions to Sean Burke, who is going to be giving the classroom today, in addition to Rui Gardner, who's the head of the facility at Memorial Sloan Kettering Cancer Center, and myself, Kathy Daniels. And we'll be distributing these slides to everyone after the classroom is complete, in addition to a recording of the session. And just one last quick comment. I want to make sure that everyone's aware that you can go ahead and use the chat function as Sean's giving the classroom. If you have any questions, anything's unclear, both Rui and myself will be monitoring that chat and either answering questions ourselves or interrupting Sean to answer some of those questions. So please feel free to utilize that. And with that, I'm going to pass it along to Sean and say thank you to him for being so wonderful and helping us out with these classrooms.
Yeah, thanks, Kathy, and thanks again, everybody, for joining us today. Let's see if I can get my screen shared here, and can you see that coming through?
Yep, we're all good, Sean.
All right, perfect. Yeah, so again, thanks for having me in for, I guess, session number 4 here today to talk about proliferation and cell cycle analysis with FCS Express. And kind of like we've done the other sessions, I'm going to make this a bit of a practical approach for performing proliferation analysis. We're going to go over that first. I'm going to give you a kind of very brief background on some of the theory behind it, and then we're going to jump right into setting up some proliferation analysis in the software. And then we're going to swing back and spend a little bit more time talking about cell cycle analysis, giving you a little bit of background about how it works and why, and then we'll go over how to perform some of that in the software. So let me just launch up FCS Express here, and again, we're going to be focusing on proliferation analysis and cell cycle today. So if you see me do some things in the software, like creating gates and inserting plots, so you have some questions about that, you can kind of save those questions, for, maybe go back and take a look at the more basic overview. I'm going to be kind of skipping some of those things, but we are going to go right into the nitty gritty here. So I was taking a look at Kathy's post-it presentation there, which looks like it gave a really good overview of some of the basics for proliferation analysis and essentially how this is working. But as a kind of basic primer, essentially what you're doing with proliferation analysis is you are loading up cells with an amount of dye. And that dye can be CFSC, it can be cell trace, it can be PKH. There's a number of different types of dyes. They're different colors, they have different toxicities, the cells. There's a lot of different things going on with them. But in the end, what you're looking to do is when you load up dye into a cell, when that cell divides, the second generation should have half as much dye as the first generation. And as that cell divides again, you get half as much dye. And what that kind of looks like in a perfect world is, if we imagine this little kind of plot down here, this image that I have, is that the starting generation will have an amount of dye. And then imagine this is FTSE signal on the x-axis. That will again, half to the next generation, and half and half and half and half again, until essentially you run out of dye or your cells stop dividing. And that's also kind of one important consideration is, when I say cells run out of dye, there is a limit to, how many generations you can detect when you're doing this sort of thing. So if you have this kind of grand plan of following cells for 20 days and looking at 20 cell divisions, it's probably not going to happen for a few reasons. One of them is, a lot of these dyes are actually cytotoxic to your cells. So, if you load up your cells with CFSE, you might be able to get a very, very bright starting generation and be able to track more generations. But if that CFSE, because you put in such a high concentration, kills those cells, or another thing that can happen is maybe it inhibits some sort of the biological function of those cells that you want to monitor, it's really kind of counterintuitive to what you're doing. So again, I think the core facility is probably a really good resource to go and talk about how to perform these assays. I know that Kathy had some references to Paul Wallace's work up on that post-it note, and a lot of his work, he's kind of one of the pioneering people doing this kind of work years and years ago. You can go back and kind of look at the literature for that. But what this data looks like in not a perfect world, kind of more of a biological world, so to say, is something more like this image in the middle. So instead of having nice discrete peaks with, say, a CV of 1 at every peak, you actually have overlapping peaks, right? And one of the reasons that we use proliferation modeling instead of just kind of saying set a gate on what looks like a population is because you want to look at the overlap of these peaks. So if this is the starting generation, some of the starting generation actually appears in the second generation, and then some of that appears in the third generation. And what proliferation modeling tools do is they allow you to kind of use a mathematical model and go in and deconvolve those peaks and deconvolve cells that are falling between different generations. The other additional consideration is that you're also going to have cells that appear in this kind of green peak that have either not taken up dye, or they've divided to the point where there's no dye left in the cell. And essentially that's what we're going to be considering a background population, or an unstained population. So you're going to see this in just a few minutes when we load up some data and we take a look. But again, this is kind of the theoretical model, but in reality, things kind of look like this. And actually, in reality, things can look a lot worse than this, honestly. Proliferation assays are notoriously technically difficult to kind of pull off. You're waiting, 4, 5, 6 days before you can even go analyze and bring your cells to the cytometer and look at it. So if you made a mistake on day 1, you might not find out till day 5, and then your week is gone. You have to start again. So again, I do recommend, if you have folks that you can talk to at the core facility before you kind of jump into this stuff, go speak with them, and maybe they can, help show you some of the kind of common pitfalls that people run into before you, kind of waste a week and realize the data doesn't look so great. So let's jump right into this. What I'm going to do in FCS Express here is open up the data list. And in the data list, I've already actually loaded up a few different data files. And essentially what I have here is an unstained control. So I believe we're looking at cell trace violet in this particular proliferation assay. So this has no cell trace violet. These were just run through the cytometer with no dye. We have a control where the cells have not proliferated, so having a non-dividing control is another important control to have when you're running this type of experiment. And a third control we have is a stimulated sample. So we add some sort of reagent or something to these cells that we know is going to activate some sort of proliferation in them. So that way we can be sure that our proliferation assay is actually working. And then after that, we have a sample, right? So something we've added some sort of reagent to that is actually going to cause these cells to divide. Maybe it be some sort of activation, IL something or other, whatever it is you're going to kind of measure. And in this case, we just have one sample, but you can imagine if we had 10, 50, 100, 1,000 samples, we could add all of those to the data list. So the place I'm going to get started is loading up one data file into FCS Express. And it doesn't really matter which one I start with here. I'm just going to load in the unstained control. The first thing I really want to do is set up some gates because I do want to make sure that when I do a proliferation analysis, I'm only looking at what I know essentially is a cell. I don't want to be trying to model debris and things like that. So I'm going to create a gate here. I'm going to call this cells. I'm going to drag and drop this out, and I'm going to look at a live dead discriminator, which is our cytox dye here. So anything that did not uptake cytox dye is going to be considered a live cell. And from there, I can take this out and I can start looking at another plot. And you can see that the other two dyes that we have here is cell trace violet, and we have a CD4 dye. Now that I have my unstained control in here, I do think it's useful to kind of start looking at my other samples. So if I need to kind of adjust gates for these, include some of this. This is going to be my undivided control, which is why we see a cell trace just appearing up here in the upper right. I have another sample where I have some more events, and we can see that we're starting to see some divisions, some populations kind of appearing throughout the cell trace dye. And again, if we look at our sample, we have the same thing. So again, first thing to do, come in, set your gates, make sure you're looking at the cells of interest. Now I can kind of take this plot and move it aside. The other place I like to start with proliferation analysis is inserting some histograms for my data and taking a look at the cell trace or whatever proliferation parameter it is I'm interested in, and seeing how that looks just simply in a histogram. So one of the things you can do, you want to make sure you get your gates applied to that. You can see how that really kind of cleans up the data. It removes a bunch of that stuff that was kind of down on the low end, which is debris and things that we don't want to include in our proliferation modeling. And again, we can kind of navigate through here and see how this data looks. So what we're going to be trying to do in our proliferation modeling is essentially figure out where cells that are undivided are located, which is generally going to be on the high end over here somewhere. And then from there, we want to kind of somehow determine where our individual next generations are falling. So in a perfect world, you're going to see some nice data that kind of, looks like this. You're going to have little peaks here, and we're going to be able to assign a peak to a generation. So we'd have a generation, starting generation somewhere up here, maybe the second generation here, third, fourth, fifth, sixth, so on and so forth until cells have stopped dividing. Again, this is a perfect world. The data doesn't always look like this, and it may not look like this on your first go around, but again, if you keep with it and talk to some of the people who know how to put together these assays, your data can look like this, right? So don't be discouraged if it doesn't look like that immediately. Now, I have one histogram in here, but I also want to start modeling proliferation for these. So I'm going to come up here to the Insert Proliferation icon, and I'm going to insert a proliferation plot down here. And I'm going to switch over to my cell trace parameter. And you can see here that in this particular plot, we have a number of different overlays. We have that red line, we have some little green lines, we have some other little histogram peaks in there. And this is FCS Express trying to fit this data to the proliferation modeling tool that we have in the software. But again, gates really clean this stuff up, right? Right now, it kind of looks like nonsense, but if I apply a gate to that and I start looking at my cells, and if I apply another gate and I start looking at my live cells, then we start deconvolving these different generational peaks. So each one of these peaks represents some sort of generation, and each of those is also going to have per generation statistics associated with it that we're going to take a look at in just a minute. Now, the other thing that's useful for these proliferation plots is knowing where your starting generation and background noise is. And one way that you can do that is if you right-click on a plot, there's options here to show the undivided cells peak, and you'll have a nice little line that appears there. And you can also show the background. Now the background, it's going to be hard to see. It's actually set at 0 by default, so it's way down here. But one thing you'd notice is if we go to our unstained control, is that our background isn't really at 0, right? Our background is up here at around 100, somewhere between 100 and 1,000. So it's not so useful to use the default of having a background of 0 when you really do have a known background for an unstained control. So there's a number of different kind of options that we can set up in our modeling to force FCS Express to use our controls for that modeling. But to do that, I want to get this layout in FCS Express set up so that I can actually use those controls, and I'll go in and I'll show you how to set those things. So generally what I like to do is I like to take my unstained control and drag it onto one of the histograms I have created and say, just show that control in that plot. And again, you're going to want to have an unstained and you're going to want to have an undivided control, which I do have. And I'm going to put my undivided control in this other little histogram. And then I'll be able to have a kind of point of reference for how this assay is working, an undivided control and an unstained control. Now, the information that I want from these is essentially the median values for the events that fall within these peaks. And again, one of the ways to do that with, markers on histograms is one way to pop that in. So if I set up some markers here, what I'll be able to do in just a few moments is be able to grab the MFI values or the medians from those and use those as the control peaks for my modeling. So that being said, I'm going to kind of get rid of my data list. I'm going to get rid of some of this extraneous stuff for you here. And we'll get into the meat of how these plots work. So if you double click or if you right click on a proliferation plot and choose format, you're going to pull up the whole formatting screen in FCS Express here, and we know to default to proliferation fit when you look at these. And there's a number of different variables and options that are in here. So one of the things that's in here is the maximum number of peaks. So we always default to 10, so we try and fit everything that's in there. But if you do have, less proliferation, you can change these maximum numbers of peaks, and it's going to kind of remove, things that are down on the low end. So if you run a proliferation assay for, say 3 or 4 days, it looks like this one was probably run 7, 8, 10 days. If your data is only going out to 4 or 5 generations, you might want to kind of change that. There's also a number of options here that change how we're fitting these individual little peaks that are in here. So generally, one of the things that we take into account is the CV or the spread of these individual generational peaks. So there's a number of options here where if you want to use a fixed CV, instead of letting this float and using kind of the default options, you can set a CV. And you can see what that does here is it essentially just lowers the CV of these peaks that it's finding to whatever you put in. The other thing you can do is you can say use the same CV for all peaks. So if all your peaks are kind of equally distributed in terms of spread, using the same CV for these might be useful. But again, if they kind of differ, if you have some peaks that are bigger than others, if you uncheck that button and let those things float, let me kind of come back over here, it'll give you a kind of representation for the data that way. Now, the other thing that's here that's pretty useful is the peak ratio. So again, in a perfect world, the peak ratio between the starting generation and the next generation is going to be half, because half the amount of dye that starts in your starting generation is going to end up in the next generation, so on and so forth. So, with this data set, again, it's a really nice data set that somebody collected. The peak ratio is 0.51. That's pretty darn close to 0.5, which is ideal. But if you find that the peaks are kind of more separated or closer together, you can come in here and you can say use some sort of fixed peak ratio and come in and enter any value that you want, right? And the one thing that I should point out is that with any of these modeling tools, the defaults that we use in FCS Express are based off of the theoretical model. So if you come in and you find that you have to change many of the variables that are in here, and you have to change them away from what the defaults are, it means that your data intrinsically is not following the theoretical model for the way that we model this data. So essentially what that means is that old adage of garbage in, garbage out. Right? If your data doesn't look good and the model's not fitting it very good to start, and you have to change a whole bunch of variables in here, and you have to change it to some extreme levels. I mean, if you have a peak ratio of 0.75, you're not following the model of what's being expected. In those kind of cases, you generally have to go back and probably rerun the experiment. Right? But if you come in and everything is kind of looking more or less pretty good when you look at it, and it's following essentially a lot of the defaults in the software, you're going to be pretty confident that your data was acquired in a good way. Now, one thing that we have here is use a fixed starting generation. And this isn't really a variable that, if you change it, you're getting away from the theoretical model. What this is doing is FCS Express looks at the data and it tries to find where a peak generation is. But if you have controls like we do over here that can tell the software where that peak ratio is, essentially what you can do is you can remove that value and you can check that box and you can say use a token, which is essentially a statistic from the data, and we can go in and we can say, all right, well, I have a plot and that's this histogram here. It's got a marker on it. It's marker 1, and if I use the median for that marker, that's going to be my fixed starting generation for this entire experiment, because that's the control that I'm using. Now, we default to show these values in gray here as a kind of long line of text describing what the token is. But if you right click on it, you can show the value, and you can see that that value in this case is 283,5642, right? So that's the median for this marker value that's there. And again, instead of using a background noise of like 1 or 0, well, we have a control, so let's use it, right? So we're going to go in, we're going to say insert a statistic for our other histogram. And again, for this one, it's going to be M1, and we'll choose the median or the MFI for that. And again, what we'll do, I like to show that value so I can see what it is. And right, it's at 1500 instead of being at 1. Now, because these values are linked up to these markers, if I change this marker position for some reason, what's going to happen is FCS Express is going to update that marker position on the plot. It's going to update the modeling based off of where that marker is. So I'll make some extreme changes here, and you can see that we're actually modeling this stuff in real time as we move that marker around. And in terms of speed, that's a pretty impressive thing. But again, what we're doing now is we're using our controls to set where our starting generation is and where we know that cells that have no dye dilution are going to end up with in the end. Now, I'm going to make one change to these two plots here, because you're going to see in a few minutes I'm going to batch process through these samples. So one thing I wouldn't want to happen is when I'm changing data between plots, I don't want these data files to change. So there's an option here in those data files where I can come into the overlays and I can say, when I change data files on everything in the layout, ignore these, right? That means when I run a batch, when I export this data, when I go up here and I look at the data list again, and I click on, say, another sample, every plot in the analysis will update except for these two controls, right? And that's pretty important because we want our controls to stay in the same position, but we want our data files that we're going to analyze to kind of float around as we're looking at them. And when we run a batch process in FCS Express, it's going to work very much the same way. Just in the interest of time today, I'm not going to set up the batch, but essentially you'd come into the batch, you'd say export to PowerPoint, export to PDF, export to Excel. And what FCS Express is going to do is switch between the data files, export all that information out to whatever format that you want. Now, the kind of one last thing we didn't talk about in here yet is getting some statistics out of this, right? I'm going to kind of move some of these plots out of the way so I can make some more room to put in some statistics for my proliferation. We'll get these right over here. They're not super important now that I have them set. So I have this proliferation plot, and if I want to get some information about this, what I can do is right-click on the plot, and in statistics, I can use proliferation fit statistics. Pop this down here before I talk about it, and I can use my proliferation population statistics, and I'll put this over here so we can see it. So what?
Sorry, I just wanted to interrupt really quickly with a question. So one, just a reminder to everyone, they can ask questions as we go along. And then two, I was curious if there's a minimum number of events in order to be able to detect a population. So is there like a threshold to be able to classify it as a certain generation?
Yep, and somehow I knew that question would arise somehow. And it's going to be valid what I say for this and for cell cycle analysis. Essentially, I don't know what the minimum limit would be, right?
Okay.
Some people, some people say they can get away with a few hundred. Some people say they need 10,000. Some people say they need 50,000, right? Maybe you need a million. I'm not quite sure. Really, what you need is whatever minimum it takes to model this data correctly. So I would err on the side of caution and collect more events if possible. When you're kind of setting up for these type of assays, you're generally not coming in here and just running the assay one time and praying that it works, because again,
Yeah.
it's a very technically challenging assay to run. So as you're running this assay and as you're developing it and setting it up, you can kind of get an idea of how many events you're going to need. And if you ran it and like the results looked kind of, wonky, and you need to rerun the assay again next time, well, maybe you go in and you double the amount of cells that you're working with. So, depending on what you're looking at, and also if you need rare cells, right? The one thing I didn't show yet is you can, you know, apply gates to these and look at your CD4 positives and your CD4 negatives. Well, if you have some sort of rare event population and it's 10% of your cells, you might need to collect 1,000,000 cells just to look at, 100,000 of the ones that are super interesting. So there's no cut and dry answer. But again, as you optimize these assays, as you're running them, you're going to be performing this over and over again. You'll kind of get a better feeling for the way it works. If you have really perfect data and your assay is like super well optimized, you can get away with less events, right? But if you have like kind of big spreads in your data, big CVs, you're going to need more events to kind of offset, that kind of error.
Okay, great. And then there was one more question that came through before you dive in there on the statistics. So there was a question, are there any diagnostic fit tests for proliferation modeling like T2? I'm not familiar with
Mhm.
that, so passing that along.
Yeah, so for the proliferation modeling, we do give some chi-squared values here. When we look at the cell cycle analysis, I'm going to come back into that as well. I think for cell cycle analysis, it's a little bit more important to try and determine which fit is best for your data, and we're able to show you all the different chi-squared values for that. But again, I think that's a good tie-in to kind of talking about some of these statistics. Um, just in the interest of time, I'm not going to go through all these statistics in great detail today, but what I think should be noted here is that these are the statistics that are most asked for, over the last 10 or 20 years that we've been doing this. So all the kind of things that you're going to be looking for: division index, proliferation index, the number of original cells, all that kind of stuff is going to be in here. A lot of people just publish with division index or proliferation index. If you're interested in mathematically what these actually mean, there's a lot of resources on our website. If you just go into our website and you type in proliferation in the search bar, it's going to bring you to all the information that you need. There is no standardized kind of way of describing what division index is and what proliferation index is. I know that seems a little um, counterintuitive. So when you look at different softwares, these things might mean different things. So division index and FCS Express might be what Flojo calls it proliferation index, and vice versa. But in the end, a lot of the times when people publish with this stuff, they make some sort of reference to a publication that gives the mathematical formula for what it actually means. So again, these things they're kind of technically challenging assays to run, but it is important to go back and I think look in our manual and get some more information about the mathematics behind what these mean as well. But the other kind of important thing that people generally look at is they want to know how many events are falling into all of the different kind of generations. So you can see here what we give you in FCS Express is the number of events that fall into these generations. And with all of these statistical kind of defaults, you can right click on these charts if you want to look at the number of original cells that cell came from, percentages. You can always expand out these tables to kind of look for whatever it is you need. So again, the statistics are here. All the things that you need are here. And from a practical approach, what I really wanted to cover today is kind of how to set up these plots, how to get these in here, and how to work with them. The other thing I wanted to cover with proliferation plots, because I even noticed in the little post-it that Kathy had up there, is one of the kind of common ways to publish this stuff is not to use the kind of default fills that we put in here for the proliferation modeling. And what I mean by that is a lot of times what folks will do is they'll go into the overlays for these plots and they'll assign a solid fill. They'll give them some sort of transparency. And this already starts looking a little bit nicer. But what they'll do is for every generation, say maybe generation 6, I'll come in here and I'll say, make this blue. And generation number 6 is now going to be highlighted blue, right? So you might be wondering, all right, somebody's got that really cool looking, publication, and they have all their generations colored different colors. Well, essentially that's how we do it. We go in and for all the different overlays, we assign whatever colors we want, whatever your color palette is for that day or for that experiment. We can come in and assign that. But again, the nice thing is once you have this assigned, when you save this layout, when you come back and you do this again, or if you change between different data files, all of that information is going to be retained, and you're not going to have to go in and manually assign proliferation overlays different colors. And I think the one last thing I should cover with proliferation, let me drag this gate out here and drop it, is if you need to look at the proliferation between different populations, again, in this case, I had some CD4 positives and CD4 negatives. Well, this works just like anything else in FCS Express. If I have a gate created, I'll just create a gate here, call it gate 1. I can apply that gate to the proliferation plot. And if I need to look at the proliferation statistics for my CD4 positives, well, when that gate is applied, we're now looking at the proliferation statistics for the CD4 positives. And I could create duplicates of this plot. I can look at CD4 positives versus CD4 negatives. Or if I just move this gate over a little bit for this instance, we're going to see this proliferation plot update. And now we're looking at our proliferation statistics for CD4 negatives, right? So again, I think these are some of the more basic and practical ways to work with these analysis in FCS Express. Proliferation analysis comes for free in every software purchase that we have. So there's nothing that you need to add on to the software to kind of go in and do anything. It's all going to be here. And really what I can say from a personal experience, I used to run a lot of these assays back in the day. If you can go to the core facility and you can get as much information as possible about kind of pitfalls and ways that you can optimize your experiment before you even start running one of these things, the better. Otherwise, you might spend weeks or months trying to optimize this, optimize your assays so that you can get the data to kind of look like this, to get it into some reliable context for analysis and also a reliable context for publication. But again, I know Kathy and Rui and the core facility, they have all of this information. They have years and years of experience. So hopefully they can save you weeks and months from trying to optimize these things, and that analyzing this stuff in FCS Express, as long as the data is good, is very straightforward. Right? And that's going to go with any proliferation modeling tool that's out there. As long as your data is good, these tools are going to work well. And again, as I mentioned, the other big takeaway is the more that you get away from the proliferation fit defaults, the worse your data is, essentially, the less that it's following the theoretical model for putting this stuff together. So with that, I'm going to kind of switch gears and move over to the cell cycle analysis. But I just thought I'd see if there's any questions that came through before we kind of switch gears and do that. If there's not, that's okay. All right. So what we're going to cover with cell cycle analysis is essentially a little bit of an overview of cell cycle analysis. I was talking with Kathy earlier before the call kind of got started, and it's kind of interesting. I feel like proliferation assays are a little bit trickier to implement, but easier to analyze. And cell cycle experiments are a little bit easier to implement, but harder to analyze, right? So I want to give you a little bit more basics about the theory before of these things before we get started. So for cell cycle analysis, essentially what you're doing is you're measuring DNA content. And cells need to be stained with some sort of fluorescent dye that's going to bind DNA in a manner that accurately reflects the amount of DNA present in them. So things like propydion iodine, BRDU, 7-AAD, DAPI, I'm sure you could look through all the catalogs and find hundreds of things that do this. There's new ones added all the time. But in the end, the fluorescent intensity of the stained cells has to correlate with the amount of DNA that they contain. And if we kind of go back to basic biology, the amount of DNA content that a cell has is going to double as it progresses through the cell cycle. So a cell that's in G0, G1 is going to have 2N DNA, and a cell that's in G2 is going to have 4N DNA. And essentially, that's the assumption that we're going with when we're modeling data, when we're telling a robot to come in and look at your data. Cells that are in G0, G1 are going to have half the fluorescent intensity of cells that have doubled and are G and G2. And really, everything in between there is going to be considered S phase. The other kind of cool thing, when we talk about FCS Express, these same kind of rules apply when you work with image cytometry data. I know we're working with flow cytometry data primarily, but if you do work with an endless image stream, if you're taking pictures of cells on glass, these same kind of tools can be used for that. Now, when we model these types of data in FCS Express, we use a tool called multi-cycle. And I'll tell you a little bit about that real quickly. Multi-cycle, the basic version of it comes for free with FCS Express. There's a plot type in here in the insert tab called multi-cycle DNA. Multi-cycle is a tool developed by Phoenix Flow Systems that's been directly integrated into FCS Express. And what the basic version does is it analyzes kind of basic single cycling data and doesn't take into account any sort of automated background aggregate and debris removal. And we'll talk about that in a few minutes. There is an advanced version that can analyze multiple cycling populations. So if you want to discriminate between diploid and aneuploid populations, if you want to automatically kind of remove background and debris, the advanced version can do that at additional cost. If you're working at the any sort of site license at Sloan Kettering or beyond, you can always add on the multi-cycle add-on. It's actually not very expensive to put that onto your license. And if you've already purchased an FCS Express license, it's something that can be added at cost later too. But again, what I'm talking about with debris removal, and we'll kind of come back into that, is if you're not doing debris removal automatically, essentially you're kind of setting up gates. And if you look at this plot, we can kind of see this is a G1 population. There's an S phase in between. There's a G2 over here. There's some stuff out here. We'll talk about that. There's some stuff over here. We don't know what that is. But there's ways that we can gate that out. So even if you're using the basic version, there's ways that we can do that. But if you're using the more advanced version of the software, all of this kind of stuff in blue hatching, this is things that the multi-cycle modeling tool has identified as background, as debris, as aggregates, what we're going to come back and talk about that, and it will kind of automatically remove those for you when you perform your analysis. So again, it's kind of letting the robot do it instead of you doing it by hand. There's different trains of thoughts about whether you should, let the robot do that, whether you should do it by hand. I'm going to leave it up to you to decide. There's no kind of empirical way to say one is better than the other. They both work pretty well if you know what you're doing. But that's kind of the difference, one of the differences between the basic and the advanced version. The other thing that the advanced version will do for you is if you have aneuploid populations and you have multiple cycling populations, hence the name multi-cycle, FCS Express in this tool will help deconvolve them. So in this case, I have some red hatch markings in here. This is a diploid population, but within that same flow cytometry run, that same set of cells, I have some cells in blue hatching over here in the modeling, and that's an aneuploid population. So that's a two-cell cycling population or two-cell cycling models. Multi-cycle can handle up to 3 multiple cycling populations at the same time. So again, if you're doing some kind of more advanced cell cycle work, if you're looking at more than a single cycling population, the advanced tool is for you. Or if you need that kind of background and debris removal, that tool is for you as well. So we're going to kind of continue in here through our PowerPoint and look at another example. Just again, as an overview, Those cells that are in 2N are going to be G0 and G1. And if we look at how that data looks in a perfect world, kind of like I looked at the perfect world with proliferation analysis, you'd have a peak at 2N for whatever dye that you're using. Everything in between is going to be the S phase. That's going to be this kind of variable portion in the middle. And things that are at 4 times the DNA, the dye fluorescence are going to be G2. So again, that's a perfect world, right? Now, in the biological world, things look more like this. This got a chart on the bottom right. You're going to have things at 2N, you're going to have things at 4N, you're going to have things in between, but you're also going to have stuff outside, 4N plus, 1N, 1N cells, or I put a question mark for that because what is it actually? It's probably not a 1N cell, right? But what will happen is when you perform cell cycle analysis or when you perform flow cytometry, what can happen is when you run cells through the flow cytometer, multiple cells can get stuck together. So you can imagine if a cell has an amount of dye and it's in G1, if 2 G1 cells are stuck together, well, they're actually going to look like a G2 cell, right? And if you have a G1 cell stuck to something that's a piece of debris, well, that's going to be kind of like a G1 and a half, right? So that's going to be a variable S phase looking cell. So there's a lot of different combinations that are possible due to clumping, right? You could have G1 stuck to G2. We say doublet discrimination. These things generally aren't doublets, right? You can have 2 cells, 3 cells, 8 cells. A bunch of different things can be stuck together. But what we do with our data is that we can use the different parameters in our data. You may have seen side scatter A, H, and W and wondered what the heck were those? Why don't we just have one side scatter parameter or one, parameter for everything? Well, area means when we look at the signal or the pulse signal for that cell as it passes the laser in the cytometer, area is the area under that curve. And height is the height of that signal, the intensity of that signal. And width is the width of that signal as the cell passes the laser. So what we can do is we can use those area, height, and width parameters to kind of discriminate between cells that are stuck together. So a G1 cell is going to have a low height, but it's going to have a high area, right? So 2 G1 cells stuck together. And those are some of the tools that we can use to kind of clean up this data. We call that doublet discrimination, which we have up here. And we're going to go to some data and take a look at that because that's a good place to start for performing any of our cell cycle analysis, whether we use modeling or we use some of the other techniques that I'm going to show here in a minute. So I've got a layout opening up. I'm going to pull that over. And again, this is kind of, I think, one of the places that we need to start when we do any sort of cell cycle analysis. We'll let FCS Express load this up. I think Zoom is slowing me down here a little bit, but just give it a second and it will pop into what we need to talk about. And grab this. All right. And again, we're in FCS Express now, and we do have some real data in here. I know this kind of looks like the PowerPoint, but essentially one of the places that you can start if you're not using the automated background and debris removal tools is getting this data in this plot cleaned up a little bit by applying gates, right? So this is actual cell cycle modeling being done. And you can see that if I apply my scatter gate over here, a lot of this stuff that's probably debris is being removed, right? Debris, clumps, things like that, it's starting to be removed. But what I have over here on the bottom is 2 plots looking at width versus area for my cell cycle parameter and height versus area. So if I apply my height versus area gate to this plot, we're going to see that some of this background stuff gets removed again. And again, if I kind of go further in for that doublet discrimination, some of that stuff gets removed yet again. So again, what that's going to do, it's going to clean up the data for my modeling without having to use automated background and debris removal to get rid of those things. And I did mention, we work with image cytometry data in the software. What you can see here is we have a little image gallery for some image-derived data. I know the histogram kind of looks similar, but I think this is kind of cool to get a visual of what some of these things look like, at least as they pass a camera instead of a laser, right? Some of this stuff up here, they're kind of, large things that are stuck together, they're clumped together, they're overexposed. And if we look kind of down here on the low end, again, what these things are kind of pieces of cells, debris that have gone past the camera, right? You can imagine instead of the camera going past the laser. Again, these are things that we don't want included in our analysis when we perform a cell cycle analysis. So again, that's one of the ways to kind of get the data cleaned up. Now, I promise we're going to get to analyzing some data real quick here. So, why would we use a cell cycle modeling tool instead of using something like this, where we'd say, all right, let's set up a marker and call these G1, set up a marker here and call this S phase, set up a marker and call these G2. Well, what happens in reality is biological samples or cells, they don't just, these populations don't just fall into neat little bins of everything's in G1, everything's in S, and everything's in G2. What will happen is there's considerable overlap between early S phase and G1 and overlap between late S phase and G2. And you can kind of see this in the plot down here in the bottom right. Some of this red hatching between the S phase peaks is overlapping with G2 and it's overlapping in G1. So what we can do is we can use modeling to deconvolve those peaks instead of just kind of arbitrarily setting markers on these things and saying, these are the bins that I decided on. And you might also be familiar with some techniques if you use BRDU, which is incorporated into the S phase when you analyze, when you mark these cells. Well, if you incorporate BRDU and you use the DNA stain, you can accurately identify and say, all right, these are G1, these are S and G2. But one of the problems with that is, well, what if I wanted to put in CD4 FITC in that assay? And now I can't because I have BRDU in that channel. Well, using BRDU and DNA, it's an absolutely perfect way of doing this. But if you want to free up that channel for doing other markers in your assay, cell cycle modeling is another kind of great tool for doing that. So we are going to go in and analyze some cell cycle data here. Before we do so, I do want to go back and say, that old adage, garbage in, garbage out. There's few algorithms out there that will handle every histogram, every set of data that you give it, particularly if the data is kind of noisy or if there's large CVs. That doesn't just go for FCS Express, it goes for FlowJo, for ModFit, any of these things that you're using. So you do have to make sure you bring good data to your analysis algorithm or your analysis modeling, just like we needed to do with proliferation analysis to get these things to work. So before we go in and analyze some data, are there any questions that came up? I think I saw the chat screen or something flash for a second there.
No, I think we're good on questions right now.
Great.
But again, to everyone, please feel free to use that chat function again if you do have questions.
Perfect. And I have a little link here. It's going to go and open up the analysis layout that we're going to look at. And again, we're going to go in and analyze some cell cycle data here. So again, I know there's a lot of background that came with that. Again, there's a lot of considerations that go into getting good data before you model it. So again, sorry, we have to be a little bit long-winded with that. But once you have good data, analyzing this stuff really with any of these modeling tools becomes quite simple. What I'm going to do here is I'm going to show you what this kind of can look like. Let's go in here and grab some data and load this up. We'll do a bit of a Martha Stewart moment here. You can get to see what's going to come out of the oven before we put our ingredients together and put it into the oven. So let's open these up. And what I'm going to do is load up some data, put it onto my plots here, change data on all plots, and we're going to be able to see some of this, what it looks like in real time, and I'm going to show you how to set this up. So essentially what we've done is we set up some gates. For this data set, we have a BRDU incorporation in case we want to look at that. And then for 7AAD, this is going to be the DNA incorporation parameter. Now, when we have a multi-cycle plot inserted, I'm going to come back around and show you how to do that in a minute. You can click on the multi-cycle tab, and this is going to show you all of the different kind of tools that are associated with this model and with this modeling fit. So keep that in mind when you're working with these plots. If you don't see the multi-cycle tab, you have to click on that plot. Once you do that, it's going to show it to you. But let's show you how to work this up from scratch. And again, it's pretty simple and straightforward to do in FCS Express. So I'm going to move that to my other screen, and I'm going to open up a new layout. And then I'll pull that over here, and we'll get crunching with some data. So again, the place to start in FCS Express with any sort of analysis, you're going to want to load some data into the data list. So I'm going to choose a few data files. I've got some samples here that are kind of controls and then a drug-treated sample. Again, just like we did with proliferation analysis, I want to get some data into here and we want to create some gates. So I'm going to do a little gate here and we'll call these cells. Pull this over. And again, importantly, when we're working with this cell cycle data, we want to get this cleaned up as much as possible before we analyze it. I kind of come from the camp where I like to get my data cleaned up before I analyze it instead of relying on background and aggregate debris tools. Again, they're both valid methods of doing this. This was just kind of the way that I've been trained. So if I want to do some doublet discrimination, I can come in here, kind of grab the main stuff in our population. I'm going to call these singlets. And then I've got my singlets. And then what I want to do, drag this out. And if I wanted to do my kind of typical BRDU versus 7AAD, what I'd probably do is come in, create some new gates, and we'll call this G1. Again, this is kind of working with that bivariate method that Kathy referred to in the beginning. And then I'd set up some sort of other gate here, and I'd say everything that's not in those things is going to be S phase. So pop this in here. And again, what I want to do, kind of scroll through my samples. If I need data specific gates for some of these things, I can adjust my gates, make them specific for the data files, get those things to kind of line up. And again, I'm not super interested in my bivariate analysis today. We're more interested in the modeling. So I'm just going to do this kind of off the cuff. But again, that's what I would probably do for my bivariate modeling with this, or not modeling, but bivariate manual gating. Now, again, when we're ready to analyze some cycling data, one place I like to start is to insert a histogram and go over to my DNA die and take a look at what this looks like, even before I set up a multi-cycle plot. And again, what I'll do here is I'll just apply a gate. We'll see how that kind of gets cleaned up. We'll apply our singlets gate. Again, see how that kind of gets cleaned up. And if I'm happy with the way that this histogram looks, then I'll probably proceed to go through and create a multi-cycle plot. And again, I do that through the Insert tab, Multi-Cycle DNA, kind of just like next to where the proliferation plot is. I can draw that, and it's going to prompt me for which parameter I want to plot, which is going to be my 7AAD area. And that plot's going to be inserted, and FCS Express is going to try and model it. Now, again, what I want to do is get my gate applied to that data. And what that's going to do for me is clean it up. And you can see in this case, we've very accurately gone in and identified G1. Everything between that G1 and G2 over here is going to be S phase. And we're kind of showing that in the plot. I don't really need to use my histogram anymore. I'll just kind of get it out of the way so we can focus more on this. But just like the proliferation plots, these multi-cycle plots have a number of formatting options. So if I right-click and I choose Format, there's a number of ways that I can come in and I can look at the different variables associated with these plots. Now, by default, FCS Express is going to try and fit things automatically. So automatically detect fit type is what that means. And if I uncheck that button, essentially what I'll be able to do is come in and change any of the variables that are here to try and make kind of better assumptions about my data. And again, what these things do here is if, we need to kind of force it to use some sort of CV value, if we want our CVs between G2 and G1 or the kind of width of those peaks to match, we can force it to that. If we wanted to kind of manually type in where we think G1 should be and where we think G2 should be, we can go in and we can manually type that stuff in. But again, just like I mentioned with the proliferation plots, the more that you change these variables, the further you're getting away from the theoretical model of what this stuff should look like. So if you find that you come in and you see that your G2 to G1 ratio is 1.5 or it's 4 or something crazy, again, G2 should be twice as much as G1, so it should be somewhere around 2. So if you find that that number is really kind of off and you have to come in here and check this box and manually change that value to something else, well, you probably need to go back and rethink how this data is being collected, how it's being acquired, how it's being put together from an experimental perspective, and optimize your assay accordingly. So again, if you come in here and these things look all crazy and your data doesn't fit any of the kind of models that we have, you generally want to go back, revisit and take a look at that. Now, when we click on the multi-cycle tab, a number of these options, auto-fitting, the kind of cycle to use here, you'll find them up in this formatting tab for multi-cycle, or you'll find them over here in our kind of formatting tab there. You can use either one. They both work kind of interchangeably. And to get some statistics, I'm going to right-click and choose statistics, put in some DNA cell cycle statistics. And I'm going to put in also our DNA experiment statistics, get these inserted, and I'll come back and talk about what they mean. And then also our model summary statistics. All right. So when we model this data, there's really 6 different ways that multi-cycle can model how this data looks inside your multi-cycle plot. And if we click on these different models, you may see the numbers change. You may see the model look a little bit different. Depending on the model that you choose, you can see here model 5, the S phase looks like it's decreasing here. That's a very different type of model to use for this data. There's different model types that you can select, right? And you might be wondering, all right, well, what's the best model type to use for my data? And that kind of comes back to the question that was asked about the chi-squared statistics. So what we'll do is for your data, we will, for this, again, the statistics, the DNA model summary statistics, we'll show you the chi-squared values for all of the different models that are available here. So when we click on this and you see model 1, 2, 3, 4, 5, 6, that corresponds to models 1, 2, 3, 4, 5, 6. And these are kind of a starting place to determine which one is going to be the best fit for your data. So generally a lower chi-squared value means a better fit, right? So if I look at the one that's 1.92, I can take a look at that. And in the end, it's kind of up to you as the researcher and as the scientist to go in here and make a determination of, what model is fitting this data best. And you might be wondering, like, oh, well, this is supposed to be a modeling tool and it's supposed to tell me what works best, right? Well, it's making estimates based off your data. And again, in the end, you need to come in here, you need to look at this. You can look at the chi-squared values, the kind of give you some sort of indication about what's fitting better. But if you look at this data and you say, all right, well, this is much more about what I expected to get from this data, and even though it has a 3 chi-squared value as opposed to 1.92, it doesn't mean that you shouldn't choose this, right? If it makes sense for the data, it makes sense for the experiment that you're working with, then that's what you're going to be looking at to use for this. Now, the other thing I wanted to point out is this background and aggregate debris removal. So in this plot, I have it gated. I'm going to take the gate off so we can see what this kind of looks like. And what we're going to find is that if I have that gate removed, and I kind of zoom in here on this axis, what we're going to see is that there's some blue hatching here, right? And what that blue hatching mean is that is what this multi-cycle advanced tool is determining as background and debris, right? You can see that we have a BAD value of 4.47. So that's the kind of metric for what this tool is automatically determining to be background and debris. So if you want to use that, you can. But again, if I want to apply my singlets gate and ignore background and debris, or you can even use them in conjunction. If I applied my singlets gate, that BAD value goes way down because we're actually manually removing a lot of that background and debris. That's a fair way to do it as well. Okay.
We actually, sorry to interrupt. We actually have a question here from Shannon. She was asking if you're comparing samples, should you use the same model for each sample, or the model which gives the best chi-squared for each one?
Yeah, it's a very, I wish I had a yes or no answer on that one way or the other, like what you should use. Really, with this type of modeling, you do need to look at your data and you do need to kind of look at these plots one by one, right? So just because a decreasing S phase works best for my cycling sample, I can see here it doesn't work at all for this one. Or I think it was the drug treated where you're using the decreasing S, right? So you do need to kind of go through these, and you do need to look at them one by one and kind of determine, on each individual sample, what's going to be the best particular. Get my gate applied here so I can get this kind of updating and working a little bit better. You do need to go through and you do need to identify, essentially what's going to be the best for each of these. In some cases, I look at this one, the chi-squared values are pretty much all the same. So really, any of these could possibly be useful. I wish I had an empirical answer and could say, well, when you're looking at a cell treated with this drug, that particular, modeling is going to work the best. But unfortunately, it doesn't quite work that way.
And I could just add in a quick comment there from a technical perspective. You want to be sure that when you're carrying out the staining and you're starting this experiment, that you start off with the same number of cells, that you're going ahead and do counts before you go ahead and do your cell cycle, to make sure there's consistency across. Because I think oftentimes people might start off with a wide variety of starting cell numbers without realizing that you should be consistent across, and that could cause huge shifts in data as well.
Yeah, that's a really great point. And that goes for a lot of these modeling tools, even transformation tools and things like that. You want to compare apples to apples, so to say. So again, this is how we kind of put together this analysis. All the statistics you're going to be looking for are here. Our model summary statistics are going to be here. The only kind of additional thing that you'll find is one of the options here is our DNA experiment statistics. And this is essentially telling you, giving you some sort of indication about how good your fit is. So if you come in here and it says multi-cycle found all sorts of errors and your S-phase confidence is bad or unacceptable, you're going to want to go back and revisit that and see, all right, is there a different model I can choose that's going to give me some sort of interpretation for this? You can see here it does say a guideline only, right? All of this stuff, they're all guidelines. The modeling is just a robot that's going in and trying to interpret your data. Never trust a robot, right? Always trust your experience as a researcher and a scientist to go in and look at this and make sure that that data makes sense. But I do want to show you two more things in just a few minutes here. I know we're going just a little bit long, but hopefully you can stick with me here. If we go into the overlays, just like we did for proliferation analysis, if you want to get these things kind of more publication ready, the same thing applies. We can go in, we can add some sort of transparency, we can add some sort of solid fill, and if we wanted to come in and change the colors of any of these things, like say the S phase, we'll make it blue. Again, you can make these look like whatever you want and make them very kind of presentable and publication ready. And when you go through and you run a batch process on this stuff, all of those results are going to come out as publication ready results. Now, the one additional thing I'd like to do here is a little bit of a lead in also to next week's talk about spreadsheets and charting and working with things, is one of the kind of useful tools I find for working with cell cycle analysis in FCS Express is working with pie charts. Again, next week's talk, we're going to cover more about the specifics of working with spreadsheets and pie charts and things. But if you insert a pie chart, it appears blank. And if I wanted to insert a statistic for G1, for S phase, and for G2, I can actually just drag and drop these things into the pie chart. And then I have a nice little pie chart showing me the distribution of S, G1, and G2, right? And again, what will happen here is as I update gates, as I change data files, these things change. And I bring up the pie charts because, having three phases is a really great thing to put into a pie chart. The other kind of unique thing that we can do, kind of like that you can do in Microsoft Excel, is if I put in the little auto-generated spreadsheet for this, and again, we'll talk about that next week on that call, I can add in another series for this data. So in this case, I've got some, G1 cells over here. I can drag and drop that gate in there and say for G1, give me % of gated. I've got my S phase up here, so I'll drag that in here for another series. And again, this is really good, not just for comparing, say, bivariate to modeling. If you have aneuploid and diploid populations and things like that, you can compare them. But these pie charts can also be formatted, just like you can in Excel, to add what's called a ring parameter or an explode parameter, right? There's a lot of these kind of cool functionalities here. So by adding a ring parameter, what I'm doing is I'm comparing on the outside ring my BRDU determined values, and on the inside ring, I'm determining my cell cycle modeling values. And I think this is a really great representation, especially with this data set, to show you that by modeling, we're really arriving at very similar results as with our bivariate plots, right? Without having to add in an extra parameter for BRDU FITC or whatever BRDU channel that's going to go in, so that can free it up. Also, we're really taking into account the overlap between G1 and S and G2. And again, this is just, I think, an outstanding way to kind of show these things side by side. So also, if folks want to join us next week for the spreadsheets and chart discussion, we'll go over this in a lot more detail and a lot more of the other charts. But I think at a high level, and to get people started with working with cell cycle analysis, these are some of the basics. These are some of the practical ways for utilizing these tools in the software. And I hope you can get started on that kind of stuff with your own data sets. We do have a lot of resources on the DeNovo Software website. So written tutorials, video tutorials, we provide sample data. And in fact, we provide all the sample data that I worked with here today if you want to go in and start analyzing some of that. So with that, I'll open up the floor here to see if there's any additional questions that have come through. And again, many thanks to Kathy and Rui in the core facility at Memorial Sloan Kettering for hosting these talks, and many thanks for everybody coming out and listening to these topics for over an hour now. I really appreciate it.
Okay, perfect. Thank you so much, Sean. Yeah, I think everyone found it very useful. We have a lot of thank you messages coming through. So yeah, this was wonderful. If no one has any further questions, I think we're good to go. And like Sean mentioned, our session next week at the same time on Thursday is going to be using spreadsheets and integrated graphing in FCS Express 7. So please feel free to utilize the link, the Zoom link that you used to sign up for today, to sign up for our session next week and spread the word. We'll be sending out this recording to everyone likely later today or latest tomorrow, along with the slide deck from the introduction. And we hope everyone stays safe. And last but absolutely very much not least, thank you to Sean so much for being such a supportive resource to us during this time. And we hope everyone stays safe, and we'll see you next week.